Hi, Karunya. It is nice to see you. I got my dogs in here with me. They wanted to come into this bedroom with me. And now Kuchuk is getting all anxious and I think she wants to leave. So let me let her out real quick. Okay, you want to go? Okay. Dakota, you want to stay, huh, buddy? Yeah, you can stay. All right. Hi, Surya. I got Karunya and Surya, and I got Dakota over here sitting with me. All right. So it's just a few of us. So let's go ahead and get started. If you have any questions, you can go ahead and ask, and I will do my best. Hi, Rishik, to answer your questions. This thing's lagging big time, by the way. I think it's like a solid 30 seconds behind. Hey, Arnav, nice to see you. Hope everybody's doing well. So, you know how I've been starting these study sessions with sort of like a show and tell? I got something different tonight. I don't think I've shown you guys this. This is something that Mr. Jones turned me on to, and I wasn't really a fan of it until he offered it to me when I went to visit him one time. And it's nothing really spectacular, but it's carbonated water. That's what I got, carbonated water. And this stuff is amazing. I used to be a soda drinker until I found this. And now this has totally replaced soda for me. And I love this stuff. So good. All right, Karthi. Let's see. Surya's getting going. Will there be A-level questions on the quiz? Karunya, no. There will not be A-level questions on the quiz. All right. Surya, you're asking when is spliceosome occurred? I think you're asking like when or where. Surya, can you be a little more specific with your question so I can do my best to answer it for you? Arnav, what does ligase use to connect the Okazaki fragments? You don't have to know anything about that, Arnav. All you have to know is that ligase connects Okazaki fragments. Hey, Tara. Yes, Tara, you're right. There will be no A-level questions on tomorrow's quiz. All right. Rishik, how do you determine if something is anti-parallel? Well, in the case of DNA, we know that they're anti-parallel, even though today in class, Ria totally disagreed, which got me cracking up. They're actually, they're parallel, right? They're next to each other just like this, but they're anti-parallel because one of them sort of goes one direction while the other goes the other direction. So they're kind of like this, right? And by direction, I mean in the 5 to 3 and 3 to 5. Okay. Hi, Kevin. Kevin, I like the spelling of your name. Who's your biology teacher? All right, let's see. Is this super laggy for you guys too? I feel like it's lagging big time. Yeah. Weird. I'm not used to it being so laggy. I wonder if it's the room that I'm sitting in. Yeah, it's a solid 30 seconds behind. All right. Keep asking away. And if I give you an answer that doesn't make sense, then keep asking until something does make sense. Rishik, why'd you have to change your little, whatchamacallit, to a Raiders logo? Come on, man. I got to change mine to a Niners one. Sort of like train tracks going opposite ways. Yeah. You can look at it that way for sure, Rishik. It would be like train tracks going opposite ways. Definitely. Why does spliceosome occur in protein synthesis? Okay, so Surya, right? Surya? Yeah. So basically, spliceosomes are enzymes that are going to cut away pieces of mRNA that we don't need, right? Those pieces are going to be called introns, and that's called pre-mRNA. So once they all get spliced out, now we got left with exons, and these exons are going to sort of be hanging out, and then eventually they're going to get tied in together. All right, give me 1 second. Give me 1 second. All right, I am back. All right. Yeah, Ruhi, it's lagging for you too? Okay. Karunya, what are exons and introns? All right, Karunya. So, hi, Jonathan. So, exons and introns, they are pieces that make up pre-mRNA, right? Exons are the pieces that we want to keep because they're going to eventually code for proteins. Introns are the pieces that we don't need, so they're going to get spliced out or cut out. And the name of the enzymes that do this is spliceosomes. They splice out the introns, remove them, and when that's done and we have pieces of exons that get glued together, now we actually have mRNA, not just pre-mRNA. Okay. Introns don't code for the protein while exons do. Yeah. So yeah, you can look at it that way. Jonathan, can you explain the different types of mutations again, please? No problem, Jonathan. Okay, so we're going to start with sort of the basics. So a point mutation, we have different types of point mutations where it happens, the mutation could happen in one point or to one nucleotide. So if you replace a nucleotide with another one, so you replace A with a T, for example, right, or a G with an A or whatever, you end up with a substitution mutation. But if you end up adding a nucleotide in there, now you're going to have an insertion, right? And then the opposite of that would be a deletion. So what you could do with insertions and deletions is you're going to cause a complete a shift in the reading frame of that sentence. So for example, in class, we talked about the cat ate the rat as a sentence. So you have the cat ate the rat. If you add a random letter in that sentence and then try to read it every 3 in a word, it doesn't make sense anymore. So that's called a frame shift mutation. So an insertion and a deletion mutation could lead to a frame shift mutation. All right. Whereas a substitution could lead to other things. So it could lead to a missense. A missense mutation is when you have your amino acid, it changes to a different amino acid, right? If you end up with a substitution that results in a premature stop codon, then you're looking at a nonsense mutation. If your mutation doesn't change the amino acid, keeps it the same amino acid as before, that's called a silent mutation. All right, so that's my spiel about mutations. All right. Yeah, Rishik, exon expressed sequence, intron intervening sequence. Yeah, I like it. Karunya, thanks again, Rishik. You guys are awesome. You always impress me with how much you help each other. I love it. Karunya, what is this? I don't understand what that picture is of you. What is that? Huh. Ruhi, same as yours. I can't figure out your. The only picture I can really figure out is Rishik's. I know what that is. Everybody else, I don't know what that is. All right, so yeah, so I think this is going to be a sort of a lagging kind of night with our video, so we're just going to have to kind of be okay with that. Tonight's going to be a lagging night, not a leading night. In this quiz, it asks, what are the 2 enzymes involved in DNA replication? Isn't there more than 2? Yeah, there's definitely more than 2. So in what quiz does it ask that? So hi, Viba, in what quiz are you talking about? Are you asking about the one that I posted where one of our own students made it? Is that it? Let me know. Karthi, does a primer determine where DNA replication starts? Yeah, a primer determines that, right? And a primase sets a primer in place. And yeah, no problem, Jonathan. A primase sets a primer in place, and then DNA replication knows where to start. So a primer is really to kind of get DNA replication started because DNA polymerase needs to start. I think I mixed up those words. DNA polymerase is looking for a primer to to start DNA replication. This lagging thing is helping me though, because usually I'm super behind your guys' questions, and tonight I'm keeping up. Karina, it's a turtle? That's a turtle? Okay. I do not see a turtle, but I will take your word for it. Ruhi, I don't even know what that means. I'm sorry. I need to educate myself on that. Rishik, primers do determine where DNA rep starts. They are placed by an enzyme by primase. Good job, Rishik. You got it. Hi, Archit. Yeah, Jonathan, it's lagging. I don't know. Is it just me? I think somebody else mentioned it's lagging for them too, but it's definitely lagging for me. Hi, Viba. Yeah, Viba, you found it in that quiz. So I wonder why it said 2. Maybe it was just asking for just 2 of the many that are in there. But I think the way that I would want to ask that question perhaps is by asking you to list all of the enzymes in DNA replication in the correct order, in the correct sequence. I think that question, I like that one a lot. Surya, how many questions are there going to be on the quiz? Approximately 30, Surya. Approximately. And some of them are, we got all kinds of questions in there. We got multiple choice. We got fill in the blank. We got matching. The only thing we don't have is A levels. Rishik, I don't know what you're saying no to. I forgot what question I asked you. Arnav, how does DNA polymerase know when to stop the replication process? We didn't really get into that, Arnav, so I wouldn't worry about that too much. Just know that DNA polymerase is going to keep working until it doesn't. There are a lot of DNA polymerases in action. So don't worry about that part. It's not something we went over. Keep those questions coming. Alrighty, let's see. You guys, I have Dakota here with me tonight. He has decided that he wants to spend the next hour with us. You meant called primase. Yes, you're right, Rishik. No worries, I got you. Yeah, I got Dakota here. Dakota is just kind of hanging out, sleeping. He wants to be with us while we're together. I will show you Dakota at some point too. All right, I think the lag is perhaps causing your questions to come in kind of late too. But like I said, it's actually sort of helping me keep up. All right. What else you got? Any questions you got? Send them my way. Tab them out and send them my way. No, Jonathan, there are no A-level questions on this quiz. Yeah, Viba, it's lagging for you too. Yeah, I don't know what's going on. Surya, it's lagging for you. I wonder if it has to do with me. Should I restart this video? Is that gonna help at all? Archit, there are no A-level questions. I think that's the most popular question of the night. There are no A-level questions on this. Thank you for helping me out, Surya. There are no A-level questions. There are none. Thank you, Jonathan. No A-level questions. Hey, Shivani. In the study guide, it mentioned why DNA has thymine instead of uracil. Okay, no, you don't need to know why DNA has thymine instead of uracil. There's just some extra information, but you don't need to know that. Cool. You're welcome, Archit. You're welcome. All right. We got a small group tonight, but that's okay. What does a covalent bond do in DNA structure? Karthi, so a covalent bond is basically going to hold the phosphate group to the sugar, the pentose, the deoxyribose, and then it's going to hold that to the nitrogenous base. Cool. Arnav, do we need to know the specific structure of the different nucleotides? Good question. You need to know that which ones are purines and which ones are pyrimidines. You need to know which ones form 3 bonds and which ones form 2 bonds as far as hydrogen bonds. You need to know that a purine can only match with a pyrimidine. You need to know that an A can only match with a T, C can only match with a G. But as far as being able to recognize the structure, all you need to know is the rings, right? That purines have 2 rings, pyrimidines have 1 ring. That's really all you need to know. Okay. Arnav, sorry, I meant nitrogen bases. So I got to go back to your question, Arnav. What did you ask earlier? Do we need to know the specifics? Yeah. I got you. No worries. Yeah, same thing. Let me know if that answer was helpful. What's the difference between covalent bonds and peptide bonds? So Roma, peptide bonds is what holds together the amino acids. Covalent bonds is a sort of a peptide is specifically for amino acids. Look at it that way. All right. Covalent bond is a very typical type of bond, but peptide is